So in the nervous system, the neuron is going to be the basic subunit as a cell. Neurons are unique in that they are excitable cells. We can also find excitable cells in the heart, but the important part about excitable cells is that because they are excitable, they can send messages. Neurons do that through a process called synaptic transmission, in which an electrical signal is sent down the neuron to release synaptic vesicles that contain neurotransmitter. But let's take a look at the general anatomy of a neuron, the microanatomy. So here we have a neuron. Based on its shape, it is going to be considered multipolar, and it is going to have a receptive region. That receptive region is going to consist of a dendrite as well as the cell body, or the soma. And here where we see a constriction of the cell body into the axon, this is called the axon hillock. This portion of the axon is very important in that it has a high concentration of voltage-gated sodium channels. And that's going to lend itself to being able to propagate an action potential down this structure, which is the axon. In blue here, we see a fatty substance called myelin. That is going to insulate the neuron and also allow for a process called saltatory conduction, which will enable a faster impulse to travel down the axon. Not all axons are going to be myelinated. For example, pain neurons. Those are going to be unmyelinated. But in this scenario, we do have a myelinated axon. These spaces between the myelin sheaths are called the nodes of Ranvier. And so, essentially, the action potential is going to jump from node to node as it travels down the axon in these myelinated neurons. When the action potential gets to the synaptic terminal here, that is where we can have the release of neurotransmitter. That neurotransmitter can be either excitatory or inhibitory, depending on the downstream structure and the receptor type that's going to be present on that downstream structure. But when the neuron is just resting, when it's not sending an action potential, there is going to be an electrical differential between the intracellular fluid, what's found inside the neuron, and the extracellular fluid. That can be measured using a device called an oscilloscope, in which we have an electrode that is present inside the cell body, intracellularly, and an electrode that is present outside of the cell. And so it's measuring the electrical differential between the intracellular fluid and the extracellular fluid. So when the neuron is at rest, we're looking at the resting membrane potential, the voltage difference between the internal environment and the external environment. And so in this scenario, when the neuron is at rest, we have a difference of -70. So the inside of the neuron is going to be more negative relative to the outside. So what if we had an upstream structure here that was going to release excitatory neurotransmitter onto this neuron and eventually propagate an action potential? That we can see through a visualization here. So we have the resting membrane potential being -70. If we have a excitatory input onto the neuron, we're going to get a slight depolarization. So the membrane potential is becoming less negative. We're going to have sodium rushing in here, and if we get to a point of -55, that's considered the threshold for the neuron, in that once the membrane potential gets to -55, it's going to reach a point of no return, in that the action potential is going to occur every time, and it's going to be stereotypical. it's typically going to have the same magnitude as well as the same duration. So when we reach this negative 55 membrane potential and threshold has been reached, we're going to get a positive feedback of voltage-gated sodium channels opening. So once some get opened, that recruits even more to be open, and that's why we get this steeper increase in the membrane potential as we go through this depolarization phase. The potassium channels that are present on the membrane are also going to be opening, but those channels are more slowly opening. So when we get to positive 35 millivolts, at this point the sodium channels, the voltage-gated sodium channels, are going to close. And we're going to have the voltage-gated potassium channels being open. And those potassium ions are going to be repelled by this positive intracellular environment. Like will repel like here because potassium is a positively charged ion. So now we're going to have potassium leaving the cell in this phase, and that phase is called repolarization. And so the membrane potential is going to become more negative again. And that's going to be due to the positively charged potassium leaving the cell. You'll notice here that there's going to be a bit of overshoot past the resting membrane potential. That's called hyperpolarization. And the reason for that is because when the potassium channels, the voltage-gated potassium channels, are closing, their velocity of closure is slower than the sodium channel. And so a bit more potassium will leak out of the cell as those voltage-gated potassium channels are closing. But eventually, due to activity in the glial cells, for example, the astrocytes, we're going to get the restoration of the resting membrane potential back to negative 70. So that is the basis for excitability in neurons and how an action potential traverses and how it is elicited.